This protocol can provide high-throughput measurements of cellular electrophysiology in a manner that befits the rapidly developing technology of induced pluripotent stem cell-derived cardiomyocytes. The main advantage of this technique is that it is less complex and less labor-intensive than the traditional patch clamp technique, yet it is able to produce equivalent results.
So this protocol can be expanded for large-scale toxicity screening programs using human-induced pluripotent stem cell-derived cardiomyocytes. For example, the comprehensive in vitro proarrhythmia assay.
To prepare induced pluripotent stem cell-derived cardiomyocyte cultures for the experiment, first coat individual 10 mm round glass number 0 cover slips with 150 microliters of 1 to 60 basement membrane matrix within each well of a 24-well plate, and incubate the cover slips at 4 degrees Celsius for 4 hours. At the end of the incubation, remove the excess matrix from the cover slips and apply the appropriate volume of cells to each cover slip. After 1 hour at 37 degrees Celsius, carefully fill each well with plating medium and return the plate to the incubator. To prepare for voltage-sensitive dye imaging, equip an inverted epifluorescence microscope with a 40x magnification, high numerical aperture lens, and couple a fast-switching warm white LED to the transmitted illumination port. Insert a simple red 660 nanometer filter into the